que vai nos falar sobre a evolução da individualidade, uma análise histórica, conceitual e experimental. Eu queria agradecer muito a Mark por ter aceitado o convite e ter passado essa semana aqui com a gente. Obrigado.
Yeah, good afternoon. Thank you for sticking around for the last one, and hopefully everyone can stay awake. It's a little cold in here, so maybe that will help. And I want to first say thanks to Charvel and Pedro, the organizers of this project. It's been an incredibly enjoyable week. Really fantastic projects, get to meet other faculty from other universities to do the evaluation of this project, which I think went very well, and I've learned a lot. I was also just really impressed with the group of students and the research projects that they're working on, so it's great to be able to get an opportunity to speak to them about their projects. And interestingly, sub-project 6, one of the projects in the larger project, is on multi-level selection, which is something that I've worked on for kind of a long time. And so there are students like Livia and Lucia and Andrea and Victor who are all working on projects with spiders, with leishmaniasis, with other kinds of organisms, thinking about these questions about levels of selection. And so today, what I'm going to talk about is this question of levels, and it's going to be more in an evolutionary context than an ecological context. So maybe at the end I'll try to connect the evolutionary and the ecological, but we'll see how that works out. I don't know if you guys saw when the U.S. had, when Obama was first elected, there was like a very famous image of Obama that said hope, and it had this kind of graphic that Shepard Fairey, who's a graffiti artist, had done. And in the same year was the Darwin year, and so this grad student in computer science made these Darwin signs. The one on the end says very gradual change we can believe in, and then you see evolve, change over time, and adapt. I just think it's kind of cool because it makes a connection between science and politics, and also science and art, and so I think this whole idea that you have as part of one of the themes of the project, these applications of science and this sort of interaction of science with the broader society is represented on this title page in that way. So here's what I want to try to do this afternoon. We'll see how successful this is, but the outline is to sort of first provide you a little bit of a historical account of this theory of group selection, which again the people in sub-project 6 are well familiar with. So I'll try not to bore you guys. Bear with me. I hope it's not too redundant for you. And then talk about what I see as a contemporary shift in the way that this debate is taking place, and what's actually finally, I think, maybe moving this debate forward a little bit, at least potentially. And then finally, I'll talk about an experimental experimental investigation of multicellularity that I've been working on for the past couple of years with some colleagues of mine and some students, graduate and undergraduate, at the University of Minnesota. That, again, in the same way that you guys are doing these very synthetic kinds of projects, projects that draw together sort of the applied, the theoretical, epistemological, conceptual, and the social. Let's see. Here's I know you can't read this, it's not necessary for you to read it, but let me just quickly show you the curve. Most of the time when historians do history, they write history, but here, a couple of years ago, some graphic artists made this curve of group selection. So this is history down here. You start in 1859 with Darwin, and you come all the way up to 2000s. And you can see this is sort of the popularity of the idea. So here you have, if this is a theory that you scribble on a napkin, this one you discuss with your colleagues, this one gets its own conference. So as the theory is getting sort of more popular, maybe all the way up to one for the history books, right? So, and this is how group selection does on this metric, not very well. So Darwin starts out a little bit, okay, it goes up into sort of the teens and 20s, and I'll talk a little bit about this. This has to do with Peter Kropotkin and Samuel Morton Wheeler, an entomologist and ant guy. And then in the 1960s, it sort of goes below down here where it's just wrong. Right? So it goes from wrong and up again, sort of looking pretty good, and then drops off in the 70s. This is around the time that Ed Wilson published his sociobiology. Oh, sorry. And then, finally, it's sort of climbing back up again. And here you have the publication of David Sloan Wilson and Edward O. Wilson, not related. Two biologists talking about rethinking the foundations of sociobiology. So, but now I'm going to actually walk through some of this. And most of the time when we think about Darwin and we think about evolutionary theory, we think about natural selection, Darwin's mechanism for change, acting on individual organisms or traits. But it's the case actually that Darwin had a slightly more open mind than we tend to give him. And here are just a couple of examples one from The Origin and one from Descent of Man. So in the first one, he's talking about social insects, and he says, we can perhaps understand how it is that the use of the sting, talking about the honeybee, should so often cause the insect's own death. For if on the whole, the power of stinging be useful to the community, it will fulfill all the requirements of natural selection, though it may cause the death of some few members. So this is sort of the beginning of the discussion about altruism in biology, which becomes a very controversial component of the whole debate over group selection. So here again, in The Descent of Man, let's see, it's 1871, so it's like 12 years later, 12 years after The Origin, he publishes Descent of Man, where he's talking about humans now and human evolution. And he says it must not be forgotten that although as his standard of morality gives but a slight advantage to each individual man and his children over the other men in the same tribe, yet that an advancement in the standard of morality and an increase in the number of well-endowed men will certainly give an immense advantage of one tribe over another. So here you have either cultural group selection or just straight group selection. Now, by the end of the 19th century, so Darwin publishes this in 1871, by the end of the 19th century, a fair amount of sort of consolidation had been happening, and the way that Darwin was being read and understood was not to pay attention to this kind of thinking, or this part of Darwin's thinking, really to pay a lot more attention to competition between individuals as being the driving force in natural selection, except for some folks. So one of those people was the Russian naturalist Peter Kropotkin. Kropotkin published a book in 1902 called Mutual Aid, A Factor in Evolution. And he essentially argued that most animals are social. And that, in fact, though competition was very much a part of the evolutionary process, so was cooperation. And so in Mutual Aid, he makes this argument using mostly his experience in Siberia. So he had done a number of expeditions in the Siberian sort of ecosystem, a rather harsh ecosystem, where in his experience, unlike Darwin in the tropics, Kropotkin saw animals struggling together against a harsh environment. And his argument was those that were most adept at cooperation were the ones that were most likely to survive. So he says, in this scenario, natural selection ceases to be a selection of haphazard variations, but becomes a physiological selection of those individuals, societies, and groups which are best capable of meeting the new requirements. It operates largely as a selection of groups of individuals, modified all at once, or more or less in a given direction. And again, as is the case with evolution up to the present, Kropotkin is actually more well known in political science departments than he is in biology departments. He's known more in political theory for his anarchist work, and he was imprisoned twice, once in Russia, once in France, for organizing labor protests, etc. And despite the politics, or well, perhaps because of the politics, we know very little about his career as a professional scientist. He was also very well respected and offered a position at Cambridge University when he was in exile from Russia, which he refused because the condition of taking the professor of geology was that he forego political involvement. So there's Kropotkin talking about mutual aid in the teens and into the 20s. But there was another origin of species, or another origin at least. This was Genetics in the Origin of Species. And this was published by Theodosius Dobzhansky, a Russian immigrant population geneticist in the US in 1937. And this was one of the first sort of founding documents of the modern synthesis. So here again, we go from an allowance for some openness to the ideas of group selection to a more, the beginning of a process of moving to what we now call neo-Darwinism. But in the early versions, in the first version that Dobzhansky published in 1937, he does talk about populations as organisms, or at least populations with sort of constituent components or functions of organisms. So evolutionary plasticity can only be purchased at the ruthlessly dear price of continuously sacrificing individuals to death from unfavorable mutations. He's talking about genetic load. So populations have to carry sort of less fit individuals as their members in order to maintain variability for protection against rapid ecological change. And he also talks about what he calls the physiology of populations. So here again, it sounds like, and going back to these discussions of metaphor and levels that both Gustavo and Donato were talking about, and I think came up in Dan's talks as well, you can see where it sounds again like Dobzhansky is perhaps open to the idea that selection acts at multiple levels. So here he writes, these factors, natural and artificial selection, the manner of breeding characteristic for the particular organism, and its relation to the secular environment and to other organisms existing in the same medium are ultimately physiological, physical, and chemical. And yet, their interactions obey rules sui generis, rules of the physiology of populations, not those of the physiology of individuals. So here you might think about an emergent property, but I'm not sure that Donato would want to go that far. So that's 1937. You get the sort of, the modern synthesis, the neo-Darwinian idea gets established as sort of the standard in evolutionary biology. But group selection, as you saw in the curve, sort of never goes completely away. There are many different folks sort of working on it. In the early 1960s, this guy, V.C. Wynne-Edwards, and actually beginning in the mid-1950s, V.C. Wynne-Edwards starts talking about group selection as a way for populations to maintain themselves at homeostatic levels below the exploitation of resources in the environment. And so there's an interesting parallel with Kropotkin's work in that Wynne-Edwards' experience was also more Arctic than tropical, more temperate and Arctic than tropical. And he spent a lot of time looking at these kinds of birds, and I would draw your attention to this outcropping here. So you have this Gannett tree in Cape St. Mary. This one is white. This is where the birds are. There's a few birds here. But all of that area, according to Wynne-Edwards, could be colonized, and yet it's not. Why? Thanks. So here, technology. All of that area could be colonized, but it's not. And according to him, it's because of these group selection mechanisms that maintain these populations below the level of exploitation of the environment. As I said, this book that Wynne-Edwards wrote, Animal Dispersion in Relation to Social Behavior, was published in 1962. The modern synthesis was really just beginning to sort of establish itself, and evolutionary biology was sort of coming into its own as a consistent theoretical and empirical mode of explanation in biology, or mode of investigation in biology. When Wynne-Edwards published this paper, it seemed to be invoking these kinds of older arguments about the good of the species that biologists had really tried hard to sort of move beyond. And so the book drew a very critical reaction, especially from Some theoretical biologists, 3 of the 4 of these are British guys. There's John Maynard Smith there, David Lack, an ornithologist there, William Donald Hamilton on the bottom, and then the American George C. Williams, who's actually, I need to update that slide. Williams died a couple of years ago. But these guys responded very harshly to the theory of group selection and worked very hard to try to undermine the credibility, the application, any pedagogy associated with it. And it was sort of in the aftermath of the work developed here that group selection, the curve of group selection drops below into wrong. About the mid-1960s was when it was sort of at its worst. And it wasn't the case that this was just an argument against something. It was really the case that Hamilton, the guy with the straw hat, this guy who actually did a lot of his research here in Brazil, had developed the idea of kin selection. And kin selection became the standard explanation for the kind of social behavior, for the kind of altruism that Darwin was interested in, for all kinds of sort of group level phenomena. Another person who was sort of deeply influential here, especially in the 1970s, was Edward O. Wilson. And again, Wilson becomes very interesting later in the story, because initially he's deeply convinced by kin selection explanations of these group level phenomenon, and manages to popularize kin selection and inclusive fitness theory in a way that Hamilton was not quite as successful. This work on insect societies, and especially his 1975 book, Sociobiology, had a very huge impact, both in the biological community and in the broader culture. Sociobiology became a very controversial work. The kinds of explanations that Wilson proffered in the last chapter, chapter 26 of the book, the only chapter that dealt with humans, managed to upset any number of feminists, minorities, etc., because of the kinds of biological determinism and biological imperialism that they saw in the sort of sociobiological approach. Nevertheless, this really became the model, and it was that is the sort of the kin selection model, and the increasing focus on natural selection working not only on organisms or traits, but actually on genes or groups of genes. And for group selection, maybe the final nail in the coffin in the mid-1970s was Richard Dawkins' publication of The Selfish Gene. And here we have an explicit kind of statement from Dawkins about what he thinks about how nature works and how natural selection works in nature. So here in The Selfish Gene, Dawkins writes, "Be warned that if you wish, as I do, to build a society in which individuals cooperate generously, you can expect little help from biological nature. Let us try to teach generosity and altruism because we are born selfish." Now, that's quite a statement, and one that you could clearly argue with, whether it's a sort of empirical biological statement, whether it's a metaphor. This is something that Dawkins often claimed, that he was speaking metaphorically. But around the same time, 1974, Michael Giesland, a philosopher of biology and a working biologist, in his book, The Economy of Nature and the Evolution of Sex, says this about how the process of natural selection works on organisms. When he says, and he writes, "Where it is in his own interest, every organism may reasonably be expected to aid his fellows." So here you have sort of a reciprocal kind of altruism. Where he has no alternative, he submits to the yoke of communal servitude. Yet, given a full chance to act in his own interest, nothing but expediency will restrain him from brutalizing, from maiming, from murdering his brother, his mate, his parent, or his child. How insane is that? Scratch an altruist and watch a hypocrite bleed. So this is a very particular view of nature, one that wasn't shared by everyone, fortunately. So, there were, again, even at the sort of nadir of group selection, there were people like David Sloan Wilson developing mathematical models and arguments to maintain sort of the viability of group selection, and people like Michael J. Wade, who had gotten his PhD at the University of Chicago and worked with flower beetles that were actually part of the group that Donato was talking about, the ecologists at the University of Chicago. Wade had inherited his beetles from Thomas Park. Park was interested in group selection, so there's sort of this genealogy of the idea. And the idea sort of remains open a bit, depends on who you ask. For everything concerning population dynamics, according to Wynne Edwards, group selection is more important than selection at the individual level. For Williams, group level adaptations do not in fact exist. For Richard Dawkins, group selection has soaked up more theoretical ingenuity than its biological interest warrants. And finally, Elliott Sober and David Sloan Wilson on the bottom, group selection is a significant evolutionary force, and its products have been amply documented in nature. So, what's the deal with group selection? Again, depending on who you ask, here's Gould writing in his final book, The Structure of Evolutionary Theory, with respect to Wynne Edwards and the theory of group selection. Wynne Edwards' claim for group selection may be wrong, but I can cite few other theories presented within evolutionary biology during my career that could be deemed so challenging in implication, so comprehensive in claims, so fascinating in extension, and so thought-provoking. So maybe this provides some kind of an explanation for the persistence of this controversy or the persistence of the interest in this idea, and helps to explain why Sub-Project 6 is happening, right? But of course, scientists want to know what the right way to approach these phenomena are, whether this kind of theory is actually useful. About 13 years ago, Michael Ruse wrote a book called The Mystery of Mysteries, where he was interviewing different evolutionary biologists to ask them about their points of view on theories. And in the epilogue to the book, talking about these kinds of debates, he says, who is right? They're all right, at least as far as their theories work. And they're all wrong, at least as far as their theories don't work. But there are no absolutes against which we can declare one side right and the other side wrong. Even as we read now, some bright, and note this is 1999, some bright graduate student somewhere is probably about to make his or her name, by resuscitating group selection. So that was 1999. Ed Wilson was not a graduate student in 1999. But in 2007, Ed Wilson resuscitates group selection, which is pretty unusual. Edward O. Wilson, working with David Sloan Wilson, write this paper in the Quarterly Review of Biology called Rethinking the Theoretical Foundations of Sociobiology, where Ed Wilson says, I made a mistake, kin selection isn't really right, group selection is more explanatorily appropriate. And a couple of years later, again, re-invoking some of the ideas that were around in the 1920s, this idea of superorganisms, where you think about, and here again with the metaphor of Gaia or the planet as an organism, thinking about insect colonies as organisms. This is the helpful metaphors, it's an appropriate biological way to understand these entities. For some in group selection, it is. I think what happens here, and so the title of this slide, Major Transitions, has two meanings. One is for Ed Wilson with respect to the theory of group selection, he makes a major transition. But two is that I think now the controversy over group selection is becoming connected with this question of major transitions in evolution. So what is a major transition? A major transition is from unicellular to multicellular life, from solitary to eusocial, right? These are some of the major transitions that John Maynard Smith and Ernst Zafmari described in their 1995 book, Major Transitions in Evolution. And it's the kind of argument that I developed in my book, Evolutionary Restraints, and from 2010, and Samir Okasha, a philosopher of biology at Bristol in the UK, developed in his book, Evolution and the Levels of Selection. So that's the history. So that's part one. I'll try to speed up. Part 2 is the experimental evolution question. So 2 years ago, David Queller, a philosopher of biology, came to the University of Minnesota, gave us a talk on organismality. What is an individual? What is an organism in biology? If evolutionary biologists and ecologists, when I think Dan was talking about populations, and you need to count, and you need to be able to predict how many of this are there, how many of that are there. If we don't have enough of this, how do we get more if we need that? If we have too much of that, how do we get rid of those kind of invasive or proliferating species? This photo appeared under my door. Someone slid it under my door and had a question on it. It was written, which of these are the individuals? And so I took and drew hats on them, and I said, the ones wearing the hats. Those were the biggest ones and sort of the most defined ones, but I wasn't sure.
Did you tape it to your window after that?
But yeah. but the interesting bit is that if you go back, and then I also wrote on the bottom, where after I wrote the ones wearing the hats, I wrote this passage from Darwin. So this is from The Voyage of the Beagle, where Darwin writes, of these polypi, so he's talking about coral polyps, in a large specimen there must be many thousands, yet we see they act by one movement. They have also one central axis connected with a system of obscure circulation. And the ova are produced in an organ distinct from the separate individuals. Well, may one be allowed to ask, what is an individual? And so that's something that now sort of, when these guys had this question, they had this question because they had started working with Saccharomyces cerevisiae, which I'll talk about in a second. But now I'm starting to think, okay, so historically, how do we identify individuals? Sometimes it's straightforward, sometimes it's not so much. And this was a question that persisted for biologists, for theoretical biologists. In 1912, Julian Huxley, grandson of Thomas Henry Huxley, writes The Individual in the Animal Kingdom. In 1915, Charles Manning Child, a developmental biologist, he writes a book about individuality in organisms. And again, for some of us, for some kinds of organisms, characteristic megafauna, it's quite clear. I can recognize all the individuals in this room, it's easy. But when you have sort of colonial organisms or these kinds of things, it's maybe not so clear. You have symbiotic mutualisms, is this getting to them? Yeah. So if life starts like this, unicellular, how do we go from that to that? For a long time we thought that this would take hundreds of millions, maybe billions of years, but maybe not. Maybe it can go fast, and let's try. The idea was for us to try to figure out how this could go. So my colleagues and I took. Well, I'll talk about that in a second. You could go this way. You could take a small cell and just get bigger. And then you could start to get invagination, and you could get developments that way. Or you could sort of stick together, right? Maybe you could just not divide completely, or you could aggregate and make a multicellular organism that way. So these were the two possible adaptive solutions. We took Saccharomyces cerevisiae, a single-celled eukaryote, and decided to put it under strong selection and see if we could select for bigger yeast and see how they would get bigger. So you take the yeast and you put them in liquid medium, and then you leave them go for 24 hours, shaking in incubation. And then you centrifuge 100 grams for 10 seconds. And then you throw away the top 93%. And so what you have in that bottom 3% are the fat yeast, the big ones. And then you take that 3 out, you put it in the next test tube, and you do it again. And this is experimental evolution. You can use microbes and you can vary ecological conditions, and you can see what happens, right? And so that's what we did in 10 replicate populations, and in all 10 replicate populations, we put this stress on them, and over 60 transfers, and usually by about 14 transfers, you can see that they're developing this pellet. That's the yeast at the bottom. Those are the big fat yeast that are collecting at the bottom after you spin them down. So by the 14th transfer, you have that, and by 60 transfers, you're getting this really kind of fat pellet. And we got both phenotypes. We got some kind of big ones, the large cell genotype there. That's the normal size cell on the top. But we also got these. And I don't know, can you see those? So they look a little bit like snowflakes. That's what we call them. This is population 1, population 2, 3, 4, 5. All of them develop this morphology, right? So there you can see clearly. And so the first question was, how did they do this? Is this aggregation? Are these yeasts sort of coming together, or are they using another mechanism? So they could be aggregating like this. This is what some organisms do. Oops, like dictyostelium, a slime mold. It has a free living stage where it lives as a single cell, and then when resources are low in the environment, they'll come together. They'll form this amoeba-like superorganism, and then they make a fruiting body, and distribute the spores. The other way to do it is post-division adhesion. So let me try that again. You can see how they don't ever let go of the mother cell. So when the daughter cells, when it divides, they don't completely let go. So that's post-division adhesion. Theory predicts that post-division adhesion will be more likely because these are more closely genetically related. In fact, they're clones, right? They're the same. So there's dictyostelium with the fruiting body, and here's the way that they come together and aggregate to make the stalk, and then some make the fruiting body with the spores, and then the spores get sort of blown away, and then they start over again. Okay. What you're seeing here is a short video. It's 14 seconds long, and there's a freeze frame. These are the multicellular yeast that are in a drop of water, and they're growing. So one of the other things that you want to see in an individual is division of labor and determinate growth. So these are not aggregations of cells. They're post-division adhesion, and they're only growing to a certain size. Right? Here, let me play that again for you. You can see them. So what we had done here is broken them down with enzymes back to the single cell stage, and then they would grow up into these clusters. And then the clusters, and they're moving together here just by the drying up of the water drops. So they're not motile. That's just a result of the. And that's an overnight. That's a 12-hour time lapse, right? So that's them growing from the juvenile to the adult stage. And the question for us was, are these individuals? Have we evolved multicellular individuals? What do we see here? This is, again, the kind of question that was motivating earlier work on individuality. And this also raises a question about levels of selection, where selection is acting. Is it acting on the individual units, the single cells, or then at some point does it start to act on the group? But when it acts on the group, is the group now becoming an individual? Are you getting a frame shift in the type of individuality from a single celled individual to a multi-celled individual? So here we have Charles Manning Child. In short, the organic individual appears to be a unit of some sort. Its individuality consists primarily in this unity, and the process of individuation is the process of integration of a mere aggregation into such a unity. For this unity is not simply the unity of a chance aggregation, but one of a very particular kind, a highly constant character for each kind of individual. So we have determinate growth. That was one thing that we were sort of looking for. I'm going to show you another video. And this is a change in reproduction. So here you can see these are single colonies or single individuals, single clusters, And right? Let me try one more time. No, it wasn't going? No, let me go back. Sorry about that. It's good. Okay. So here, and now what you're going to see is that they reproduce by letting go of a propagule. It's not single cells that break off. They have a juvenile stage. So there's an adult and a juvenile. The blue is adult. Those are twins. Blue and green, adult, juvenile, adult, juvenile. So when they get to a certain size, they start to reproduce these propagules, and then they grow to adult size. What's interesting is, and actually let me skip ahead here, is that we're looking for now in these individuals some kind of division of labor. Different cells doing different things, some kind of differentiation. And we're also here talking about a different kind of reproduction. It's not single cells dividing, rather it's programmed cell death that is creating apoptosis, that is creating these breakpoints where the propagules let off. So what we think is going on is that these apoptotic cells, these orange cells here, serve as breakpoints for the propagules. And so you have this, you could have just random breakage, or you could have this programmed cell death, and where those nodes are is where you'll get these, once the cell dies, you'll get offspring. Right? And if breakage is random, we would only expect them to break at the apoptotic cell about 5% of the time. And yet we found more than 75% of the time they break at the apoptotic cell. And so it looks like this could be early division of labor, and that would be early differentiation. That would be further evidence that these are not collectives, but they're growing individuals, a new kind of individual. And what we think this does is connect, in a very interesting way, the levels of selections question, the major transitions question, questions about empirical investigation, again, connecting to this project, empirical investigation with sort of a deep conceptual framework. We realized when, those guys actually realized when they first started the project, that the empirical result would not be sufficient to show people the phenotype itself would not be sufficient. What was needed was a sort of historical and philosophical framework to explain and put into context the way that the projects do here the empirical result. So I'll just end with this. And again, make a pitch for the role of history of science and philosophy of science in these kinds of investigations. My advisor, Fred Churchill, wrote this paper called Darwin and the Historian a few years back. But I think it's important, and it's been really influential in the way I do my work, where he writes, "The fortunes of scientific theories determine the historical reading of the past. More sophisticated histories attempt to fill in the details in the context of science and produce low-level causal theories about network of events. Such efforts require the employment of all the tools available at the historian's bench. For the historian of biology, these efforts demand first and foremost an understanding of the science involved. Without this primary implement, the history of biology was at one time chronology without substance. With it, we have begun to get deeper into the subject." And I think that's the kind of work that I've seen here in these integrated projects, and that's the kind of work that I've tried to do with my team at the University of Minnesota, Mike Travisano, Ford Dennison, Will Ratcliffe, and then undergraduates, Kristen Jacobson, Mitch Hoberman, Amanda Neubauer, Jennifer Pence, and graduate students Jonathan Fankhauser and Chris Euler. So that's all I have for today. Thank you very much.
Minutes. We have 20 minutes for questions. Vitor? Onde? Você é baixinho. Você não perdeu peso. Tem que estar no processo de lento desaparecimento.
Bom, eu vou falar em português, tudo certo?
I translate. Just speak slowly. Fale lentamente.
Certo. Mark, eu queria saber sua opinião com relação a... Isso é meio devagar.
No, I followed. You want to know your opinion?
I got it.
E eu quero que você fale em português. The major transitions. Do you think that eu não sei falar major transition em português, né? Okay?
Yeah, the main transition.
Você acha que em todas as major transitions a gente teve um papel importante na seleção de grupo?
The question is your opinion about if in all major transitions have group selection played an important role?
I don't think necessarily, and I don't remember all of the transitions that Maynard Smith and Mari identified. I know for me and for this project, obviously the unicellular or multicellular is one of them, and I know that they have at least one of the major transitions about the evolution of social behavior. But I'm not sure about the other ones, but I would think that it's not the case that group selection would be necessary for all of them, even not remembering what they all are. And in fact, for Maynard Smith and Zafmari, they're not necessarily convinced about group selection. Maynard Smith never was. But I think what's interesting, and this was sort of evident in your guys' project, and I think we talked about this a little bit in the lab meeting, was the language is really changing here. People talk less and less about group selection for various reasons, and talk more and more about multi-level selection. And multi-level selection, it seems like even someone like John Maynard Smith might be okay with multi-level selection, in some sense. That makes sense?
If I just add one minor comment, that major transitions have to do with changes in the way information is stored, transmitted, and evolve. And this can have to do something to do with group selection or not, because there are several different ways of changing storage, transmission, and evolution of information. Pedro.
I will ask a very naive question because I don't know anything about this literature, but this experiment that you did, I almost cried here of emotion. You developed artificially a multicellular organism. Is it common? What?
So all right, I convinced you.
I almost cried. I must confess.
Well, I mean, so it was. So as soon as they saw those groups, they thought this could be really something very big. They were very excited, and that's when I got the photos slid under my door, and that's when we wrote the grant to continue the work, because they had sort of just done this as a side project. When they were listening to David Queller's talk, they thought, okay, he had this continuum of individuality, where he had these kind of social microbes and then colonial organisms like a Portuguese man-of-war, all the way up through what we take to be totally integrated individuals. And they thought, could we evolve a multicellular individual? Mike Travisano has been doing experimental evolution for 20 plus years. He works mostly with E. coli, but he and Will, who was a grad student at the time, was a PhD candidate, thought that yeast, because it was a eukaryote, because it had sex, because lots of things, that it could be a good candidate. And it worked. It took 4 weeks for them to get that first. And then we went out for beer, and we talked about it, and I thought, wow, this is pretty. Yeah, well. And then, and the initial response was very interesting, because not everyone is convinced that those are individuals, right? And it's not clear what it means to claim that they're individuals. And the kind of language that we used, proto-organism, proto-multicellular, we felt like we had to be very careful, and the review process. I mean, we published the paper in PNAS in January of last year. It was reviewed and sent back to us, 3 times at Nature, twice at Science, and each time they wanted actual mechanisms. They wanted us to do genetic work. There was work that we were doing, but we wanted to get the paper out, so it ended up in PNAS. But a lot of times, for example, one of the complaints that came from a lot of people familiar with yeast is that yeast, I'm going to forget the name of the phenomenon. Yeast have this ability when they're in scarce resources the cell elongates, and they make these kind of hair-like structures where they're multicellular, right? And so the first thing, it's called flocculation. So they said, the first thing we heard was like, oh, this is just like a weird flock. It's not multicellularity. But flocculation, the phenotype is totally different, and so we had to make arguments to convince people that this was not flocculation. Other people argued, well, it's possible, or actually there are people who've claimed that yeast was multicellular 750 million years ago. So we just sort of turned on an old genetic switch. It's not de novo. It's possible. We're not making the claim that that genetic machinery wasn't buried in there somewhere, right? But we felt like the phenotype was sufficiently interesting to sort of move forward. And so now we're trying to use selective pressure to see how strong the sort of multicellular characteristic is. Can we devolve them back to single cell state? We're having some success with that if we let them sexually recombine. So it's a process of convincing ourselves and then trying to convince other people that that's indeed what we have. But I mean, again, we can see historically that this is something that biologists have thought about, and you can read Kant, and Kant talks about the nature of the organism and what it means for an organism, for something to be a biological organismic system. And so they're very organismal to me. I kind of think that they should count. But I think it's going to take a long time to... The thing that's also cool about it, it's a very easy system to work. So we've written up a report for a methodology for doing this in high school labs and undergrad labs. It's a model system that now we can talk about adaptive radiation, we can talk about speciation, we can talk about development. It's a model system that has a lot of potential, and the individuality question is sort of only part of that potential.
Send me the protocol for high school. I'd like to put the students to do that. And my empirical colleagues, anytime you have a fantastic result, please keep something under my door.
I read the book about individuality and evolution of Julian Huxley a long time ago, so I don't remember exactly what he said there, but I believe that he said that evolution has a tendency of necessarily to produce individuals, living beings that are more individuated than others, than before, and he also said that it can be a criteria of progress.
Yeah.
What do you think about these two ideas of Huxley?
So I don't know so much about the first one, about the context of where he's saying that individuation is a natural part of the evolutionary process.
Individuation in the sense that evolution tends to produce living beings that they are not the words of Julian Huxley, but what he said is that evolution tends to produce living beings that are more autonomous in relation with the environment. More autonomous and independent of the contingencies of the environment. And this is a criteria of individuality, it is also a criteria of progress.
Yeah, no, I think, so I think that that's something about sort of the integrated nature of the individual. So the organic individual can sort of buffer environmental effects, right? So it's not totally subject to
Yes.
the extremes of the environment. Yeah, I remember that, and I think that makes perfect sense, right?
Yes.
In some ways. The question about progress, this was Huxley's thing. I mean, I think he again, here's a good example of someone who saw essentially political and social theory in a biological idea, in a biological theory, and as a representative to the UN for UNESCO and all of those things. I mean, he was somebody who argued that yes, evolution was a progressive process, that it was deeply progressive, and that this would be a natural kind of move up the lineage.
So, but independently of name that progress, you accept that there is a tendency in electron evolution of producing this kind of high individuation.
Not necessarily. I mean, I think for both the question of progress and high individuation, I mean, I guess, individuation to some extent you need, if you go back to the origin of life, you need a sort of a semi-permeable membrane to start, right? Maybe you have the replicating RNA out there, but it needs to be at some point enclosed, so it's not just a molecule floating in an environment. It's something that is now somehow self-regulating and then replicating. So that membrane is important, but that membrane is always permeable. There's always an exchange between the organism and the environment. So there is individuation, but there's not necessarily complexity. I mean, the biomass is still largely single cell, right? I mean, and I think now we have, so I think that's the best argument against the notion that there's some kind of orthogenetic or even evolutionary trend towards complexity. The random walk, the falling off the abyss, there may be a minimal wall of complexity that we're sort of randomly staggering away from at the single cell level, but I don't think there's a driven trend towards complexity in evolution. I think it's I think it's a random walk. Most things are still very simple. Most things are still single cell.
Mark, thank you for your talk. Part of the controversy around group versus individual selection depends on calculation of fitness, individual fitness, group fitness, and I would like in your example, the Saccharomyces, they are doing both things. Individuals or semi-individuals, if you wish, they are dividing themselves, but the groups are also dividing themselves. So first I would like to hear about the exact example. What do you think could you divide the within group or individual fitness from group fitness or new level of individual fitness? And the second question is just, do you think that, because I have seen many debates around these divisions, and one of the ideas is that, well, you can calculate fitness both ways. If you look at it as a group, if you look at it as an individual, it's just a way of calculating. What do you think about this conclusion?
Yeah. No, I think that was sort of where the argument had arrived before the argument about the value of group selection, before this kind of major transitions and the revivification with Wilson's 2007 and the 2010 paper, was that, look, we have mathematically isomorphic models. You like kin selection? Fine. Use a kin selection model the phenomena, that's fine. I'm going to use multi-level selection, maybe I use multi-level selection type 1 for this kind of phenomenon of interest, and maybe I'll use multi-level selection type 2 for that one. And I tend to think that that's okay. I tend to think that like a plurality of models in biology is fine. And as long as you have sort of the explicit explanation as to why you choose this model versus that model versus the other model. And I had to ask modelers, mathematical people, like, so, you know, if it's mathematically isomorphic, it means it's the same, right? It's the same, right? Yeah, it's the same. And yet the argument seemed like it was still going on. And they go, why are these people arguing? What is the point of this? I guess the point, right, is if you come to the yeast example, I think it's very hard to say exactly what's going on there, and we're developing models to try to figure out what we think, where selection is acting at what point in the process. So one thing that I didn't mention, but maybe it's obvious to you given that you're a biologist and you understand the experimental setup, but the good thing about these experimental evolution experiments is that you archive the ancestral states all along the way. So then you can take 10,000 generation multicellular organism and compete it against, under varied conditions, generation 10, right? And you can see how they behave, and you can calculate fitness in various ways, and then you can try to figure out, you can try to partition the fitness, or you can, and so we're not there yet. The way I think about it right now is that it's this frame shift. It's a frame shift where selection is initially acting on single-celled individuals to get bigger. And then you get this response of multicellularity, and then this seems like a case where you might not want to call it a group because it's genetically identical. But now the individual is a different kind of thing, and if there's a speciation event, if Saccharomyces cerevisiae is a single-celled and this is a multi-celled thing, I mean, something's happening there, right? I mean, I don't know.
There is a development now.
Yeah.
If one has development and the other has, or
Not.
either don't have or
Right.
has.
The single cell, so microbial people, Mike especially, will always argue, like, people think microbes don't have development, but they do.
No.
But certainly, the multicellular looks like it's got development in a way that
I would just try to, the way you calculate fitness depends on what you consider an individual. So, if you have the evolution of an individual, so you will have to change the way of calculating.
Right.
So
that's right. Yeah, and that's why, it's not just a sort of semantic argument for evolutionary biologists. They have to pick. They have to say, this is the important evolutionary unit, and this is an individual, not that collection, this collection. So Peter Godfrey Smith's book is really good on this Darwinian population and Darwinian individual.
That's the Dan.
So is there any, is there any evidence that, or are you looking for anything about that cell that dies that then cleaves those that's similar or different? Could those be mutant cells and therefore aren't the same individual in some sense?
So this was one of the earliest assays that we did, was staining them with that calcium and finding these bright orange, very pretty dead cells, and they looked initially like they might be at the center, so we thought it might be like resource scarcity, like they were getting starved in the middle. But then they started showing up in other parts of the snowflake. And so then we thought maybe it is just size and they get torsioned or something, they just get twisted and that's how they die. But we have a grad student, Jonathan Frankenhauser, who's doing some sequence work and trying to figure out what exactly is going on with them. So yes, it's an area that we're looking at, but we don't have any, we have very preliminary kind of results. I can't really speak to.
In the same text book you quoted Julian Sorel, 1912. he identified two types of individualities, spatial and individuality in time.
Spatial and
Spatial individuality, individuality in time. He said coexistent part and part that succeed each others in time. Did you find this type of, at least individuality in time, can be applied to this experience?
maybe, yeah, that's an interesting question. Maybe the question of the individual in time could be useful in thinking about the way that we've identified their sort of reproductive process. That you have this growth process that then leads to the breaking off of the propagule, and so maybe over a period of time, I don't know if that time, I think that time is pretty fixed, like how long it takes for them to grow to the adult size. So an individual in time has a sort of a lifespan, right? And so maybe these have a clear lifespan, and that might help to individuate them. So maybe in that way, Huxley's idea of the individual in time, is that what it means? Yeah.
And the part that eventually heterogeneity of
Heterogeneous part that succeed in time, eventually.
I'll make a final question then. We have Stuart Newman here with us like 3 months ago,
Yeah.
in November, no, not 4 months ago.
Yeah.
And one of the papers he gave us to gave the students to read was the Experiment Evolution Multicellularity.
Okay.
And but Stuart argues that the evolution of multicellularity, at least in animals, have to do with the increase of calcium in the Precambrian seas, which made the cells stick together,
Yeah.
and they couldn't avoid it, so have a new selective regime and molecules that were acting in the cellular organism in one way begin to work differently. And here we have, if we accept this multicellular organism, evolution of multicellular by natural selection and artificial selection. Do you think these results can?
I think according to like the contemporary literature, multicellularity is thought to have evolved at least 25 times, right? 25 different, and we're not making the claim that this is how multicellularity evolved. We're making the claim that this is, or we're saying what's significant about the result is that this provides a plausible mechanism. The big surprise for us was how fast this can happen, right? And so maybe you can get the evolution of multicellularity quickly, so maybe it happened a lot more than 25 times, and then it wasn't advantageous for whatever reason, and it went extinct. And then, you know, at some point it came back, and then it took off. So Newman's idea, high levels of calcium in the ocean and aggregation, sure. I think that's another possible account. I think what's nice about this system is we can now, as I was saying before, continue to run these experiments on the system and ask these kinds of questions. If you make the plausible hypothesis that under highly calcified seas, I mean, maybe they need to find a contemporary marine microorganism, stick it in some, you know, highly calcified seawater and see what happens. Maybe they'll get multicellular organisms. It might be super easy. I mean, Travisano, Mike has said many times that people working with yeast probably had this phenotype. Brewers, when you're making beer, when this happens, when the yeast stick together, it's a problem. So you throw it out. So Mike was like, there's probably many people who work with yeast who had this phenotype and just pitched it. Because that's not what they were looking for, that wasn't their experimental setup, that's not what it was designed to investigate. You know, this one started with the idea like, oh, well, let's see if we can get, and then when we got it, for us that meant something. But for other people, it really didn't.
This is a good example of serendipity.
Yeah.
So I'd like to thank Mark again, and to thank all of you for the patience. Agradecer a todos vocês pela paciência. queria agradecer a todos os convidados de modo geral.